Hello, can somebody confirm you can hear me?
Yes, I can.
Thank you. All right, today is August 8th, and hopefully after the lab today, I'll send you your unknown results. I've done some grading. Let me call that up and that'll help me. I have lab 5 graded for the first time, and I've got about half the people done in lab 6. I'll try and get the other half done tonight, meaning everyone's done halfway, but half the people I have not graded questions 5 through the end. I was doing that right before starting class. I was hoping to get finished today, but I did not succeed. And then I got graded the worksheet for the lectures 4 and 5. Let me remind you that you don't get a second chance in the lecture. You're only answering 1 or 2 questions. And so if you do not know the answer, you should look it up so that you don't get marked down. And I think that's about it. I might have done some extra credit assignments as well. Any questions on the grading? I'm going to shut down my gradebook now, and then I can share my screen. Let's go to the syllabus. So today is August 8th, and we are supposed to be, lost it. We're supposed to be on chapter 6. We're not quite there yet. We're a little behind schedule. Do note you have a quiz this week, quiz 3. It'll cover lab 5 and lab 6. And yes, I know I'm still grading lab 6, or at least on half the people. I'll try and get that done today. And then the quiz will be over chapter 4 in the lecture and the end of chapter 3. Any questions about the schedule? Today we'll cover lab 6 in the lab. And then we have a lab tonight, lab 7. And then on Thursday, we'll have another lab. All right, any questions about what we're doing? Let me scroll to the top. And open up lecture 5. Oops. That always pops up now whenever I open anything with Microsoft Word. We got to talking about photosynthesis, saying that it was the conversion of light energy to chemical energy. Photosynthesis only occurs in photosynthetic organisms. These organisms contain a light-trapping pigment, one that is known to you as chlorophyll, which makes green plants look green. The light releases electrons from chlorophyll, and then the electrons move about, giving the cell energy it uses to convert the chemical energy in light using an electron transport chain. And that energy is converted into ATP and NADPH. And they are usually used, ADP and NADPH, to make glucose from CO2 and water. This we call carbon fixation. And so the light, which was once the energy that was once in light, is converted into the energy in glucose. And that's the way photosynthesis has probably been taught to you before. But let me state that there are two types of photosynthesis. There is cyclic photosynthesis and non-cyclic photosynthesis. In non-cyclic photosynthesis, we make glucose. And the summary equation, which you've been given before, is 6 CO2 molecules plus 12 water molecules in the presence of light make glucose, which is C6H12O6, 6 oxygen molecules, and 6 water molecules. You do need to know the summary equation for non-cyclic photosynthesis. You do not need to know there is another form of photosynthesis, which is anaerobic. It does not produce oxygen. It actually uses not water, but hydrogen sulfide gas and produces sulfide. Like I said, you don't need to know that one. That is non-cyclic photosynthesis, which you've been taught before. There is also cyclic photosynthesis, and every organism that I'm aware of can convert light both with non-cyclic photosynthesis and cyclic photosynthesis, depending on what the cell needs. Like a green plant cell, like in the leaf, if it wants ATP, it will engage in cyclic photosynthesis. However, if it wants glucose, it will engage in non-cyclic photosynthesis. So it depends on what the cell needs. In cyclic photosynthesis, it's a much simpler reaction. You simply have ADP getting together with phosphate to make ATP. Any questions about any of that? All right, let's talk a little bit more about cyclic photosynthesis because you've never gotten it before. Let me blow this slide up. Cyclic photosynthesis happens with cyclic photophosphorylation. We've mentioned about phosphorylation is just adding phosphate to a molecule. And what we do is we add phosphate to ADP to make ATP in cyclic photophosphorylation. The photo just means that it's phosphorylation occurring in the presence of light. Light excites 2 electrons from chlorophyll. Those electrons leave the chlorophyll, and then they enter an electron transport chain. So the electron moves to this protein in a chain of proteins in a membrane. Come on, my mouse died. Then the electron moves to this protein in the chain of membrane, and then the electron moves to this chain in the protein, and then the electron returns to this protein. The electrons actually end up at chlorophyll, the molecule that the electron left. And that's why it's called cyclic photophosphorylation, because the electron travels in a cycle. It ends up where it began. As the electron moves through the proteins in the chain of proteins in a membrane, it gives this protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. Those hydrogen ions create a twofold gradient. There's more positive charges on this side of the membrane than that side, and there's more hydrogen on this side than that side. So these hydrogen ions are going to want to move to this side of the membrane just by diffusion. However, hydrogen ion cannot move through the lipid bilayer of the membrane. So they can only move through a protein, channel protein, and it happens to be ATP synthase. And when the hydrogen ion moves through ATP synthase, it gives this protein the energy it needs to phosphorylate ADP to make ATP. And that's the way cyclic photophosphorylation happens. This should be familiar to you because it's very similar to the way the electron transport chain works in aerobic respiration. And that is, electrons move through a chain of proteins in the membrane. And as the electrons move through some of the proteins, they give the protein the energy it needs to move an electron from this side of the membrane to that side of the membrane. And then that proton hydrogen gradient is used to make ATP. Any question about any of that? All right, cyclic photophosphorylation happens in a eukaryote cell in the inner chloroplast membrane. So this membrane here is the inner membrane, come on mouse, is the inner membrane of the chloroplast. And this would be inside the cell, or excuse me, inside the chloroplast. This would be outside the inner membrane of the chloroplast. The chloroplast has an outer membrane, and it would be out here. So these hydrogen ions will not go out and potentially damage the cell because they'll stay in the space between the inner membrane and the outer membrane of the chloroplast. Now, in prokaryotes, they don't have a chloroplast. So this membrane is the cell membrane. And this side is the inside of the cell membrane, meaning the cytoplasm of the cell. And this side is outside the cell membrane, which for most cells, this will be the cell wall. And so they're pumping the hydrogen ions into the cell wall. Any question about any of that? All right, that's how cyclic photophosphorylation happens. Let me go through a summary of the requirements of ATP production. You have an energy source molecule where the electrons come from aerobic respiration. This is usually glucose, but it can be any molecule of food. For a chemosynthetic organism, the electrons can come from an elemental source like sulfur, ammonia, or hydrogen gas. The point is the electrons are used to make ATP. Now, for photosynthesis, the electrons come from a pigment molecule, but initially the energy comes from sunlight, and the sunlight causes the electrons to move, and then that movement of the electrons is used to make ATP. There will be electron carrier molecules. We've talked about NAD plus and FAD. Those molecules carry the electrons in aerobic respiration. We haven't talked about NADP plus, but it's an electron carrier in photosynthetic organisms. My mouse is dying again. And as the electrons are carried to an electron transport chain, the electron transport chain makes ATP, and then the electron will be delivered to the final electron acceptor. In aerobic respiration, the final electron acceptor is oxygen. In anaerobic respiration, it's some molecule other than oxygen. It may be nitrate. Let's see, nitrite. It could be nitrite, nitrate, nitrite. It could be sulfate. It could be iron. It could be a whole bunch of different molecules. For fermentation, it could be an organic chemical compound. And then for photosynthesis, if it's cyclic photosynthesis, the electron returns to where it started from, and that would be from chlorophyll, at least in green plants. That's not shown here for some reason. I don't know why they didn't, because they have photosynthesis shown here, but not down here. Any questions about any of that? All right, that's it for chapter 5. Let's move on to chapter 6. Let me shut down chapter 5. Why did I have 2 of those screens? Don't know why, but both of them shut down. All right. Chapter 6 covers microbial growth. Learn the major goals, and this is giving you a rough outline. So know the terms, know what's a psychrophile, a psychotroph, a mesophile, a thermophile, what are hypothermophile, what's an acidophile, a halophile, an aerotolerant anaerobe, a facultative anaerobe, what's binary fission, what are reproductive spores, and what are endospores. Some of them we've talked about a little bit, like endospores, binary fission, I think we talked about. I think we talked about halophiles too. All right, 2, understand the various requirements for cells to grow. 3, understand the different phases of growth. And 4, be able to compare the different methods of measuring microbial growth. Any questions about what we're going to cover?
I'm not sure if you're supposed to be, or you're meaning to be sharing your screen, but you're not.
I'm not sharing my screen. Has that been all the way?
Just since the new chapter.
Oh, that's weird. I don't know what happened there. I shut down the PowerPoint, but that shouldn't have shut down the Zoom screen. Sorry about that. I'm not sure why that shut down. All right, let's move on to microbial growth. Microbial growth, when we're talking about microbes, refers to an increase in cell number, not an increase in cell size. Is that clear? Microbial growth refers to an increase in cell number.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in 2. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, any questions about any of that? Let me shut that down. Cells have different requirements for growth. We're going to talk about requirements for microbial growth. There are physical requirements and chemical requirements. Physical requirements include the cell has to be at the correct temperature, the correct pH, the correct salinity. Chemical requirements for growth, the cell has to have a source of carbon, a source of nitrogen, a source of sulfur, a source of phosphorus. There has to be a source of trace elements. If they require oxygen, they have to have the right amount of oxygen. If they don't require oxygen, they require the absence of oxygen, or they may require the absence of oxygen. I guess there are some anaerobes that it doesn't matter if oxygen is around or not. And then there are also organic growth factors that may be required. Like in our cells, for example, we have all of these, obviously oxygen 2, and organic growth factors. For example, vitamin C would be an organic growth factor. We must have it in our diet, or we would get sick and then eventually die. Any question about any of that? Each species has its own minimum temperature that it can grow at, its maximum temperature it can grow at, and its optimum temperature that it can grow at. The optimum temperature would be when a microbe would grow the fastest. Microorganisms are classified into groups based on the basis of their preferred temperature range. There are psychrophiles. Let me blow this one up. And they prefer a temperature around a mean around 10 to 12 degrees. The range can vary, but let's say minus 10 degrees Celsius to 20 degrees Celsius. Beyond that range, they would not grow, and they grow best between 10 and 12 degrees. That would be the temperature of your refrigerator. There are psychotrophs, and they have an optimum temperature growing around 20 degrees Celsius. That's about room temperature. Many organisms in the soil or in the ocean may grow best at that temperature, although that probably would be the ocean of the tropics, not the ocean of, well, not even the ocean of California, but not the ocean of Washington, for example. We would be cooler than that. Mesophiles are organisms that grow best around 37 degrees Celsius. And that's the temperature of people. And all human pathogens are mesophiles. So all human pathogens are mesophiles because mesophiles are the temperature of people. There are also thermophiles, and they grow best around 60 to 62 degrees Celsius. Quite elevated temperature. And that would be, if you ever see the temperature of the chicken rotating in the store, and bacteria can grow in that temperature. It'd be a thermophile. And so if you ever think the chicken looks like it looks old, you might not want to buy it because the thermophiles can grow at that temperature that the chicken is continually rotating at the store. The thermophiles will not grow in you, so they will not be a human pathogen, but they can cause food spoilage. And I guess the same with the psychrophiles. They cannot grow in us, so they're not human pathogens, but they can grow in the refrigerator or in food kept at 10 degrees Celsius or around that. And then they can cause food spoilage. And I guess I should state that's why you should throw food that has no preservative in it. Once it's been in the refrigerator for about a week, it should be thrown away because the psychrophiles can grow. And after about a week, you'll have enough of them growing that they can cause the food to spoil. Any question about any of that? Now, if the food has salt or an acid or a lot of sugar in it, the food can last longer in the refrigerator. And that could be like jelly. I don't know how long jelly can last, but it can last a long time in the refrigerator. And then ketchup. Ketchup has both salt and acid in it. So that can last over a year in the refrigerator. And then the fifth group, wasn't even known when I was taking microbiology. The hypothermophiles, they can grow best around 95 degrees Celsius. Remember, 100 degrees Celsius is the temperature that water boils at. Any question about any of that? The hypothermophiles cannot grow in us, so they don't cause us sickness. Their membranes would freeze at our temperature. And they do not seem to cause food spoilage because the Mexicans will take their corn and throw it in a volcanic vent, which has hypothermophiles growing in it like crazy, and then they'll cook the corn in the vent. I don't know, they leave it in there for 6 to 10 minutes, something like that, and then take it out and eat it. And the hypothermophiles never make them sick. So apparently the hypothermophiles are not easy at spoiling food. I'm not sure why that is the case, but it could be just that hypothermophiles are so unusual that they don't tend to cause food spoilage. I'm not saying they never cause food spoilage, and it could be that the Mexicans are used to eating them, whereas an American, if they were to eat the corn that had hypothermophiles on it, it could make them sick. I mean, that's a possibility. I don't know. I've never attempted to eat hypothermophiles. Any question about any of that? All right, let me state that the various classification systems are not rigidly defined. This is a basic organ definition. The range in particular is not rigidly defined, as well as the optimum temperature. Like mesophiles are generally organisms that grow at 37 degrees Celsius, and that would be true for something like Streptococcus pyogenes, or what's something that grows in the blood? Streptococcus meningitis. But for Micrococcus luteus and Staphylococcus epidermidis, they have an optimum temperature around 30 degrees, not 37. And that's because they're growing on our skin, which is typically cooler than inside our internal body. And their optimum temperature for growing is around 30 degrees. And yet we still call Staphylococcus epidermidis and Micrococcus luteus, and these organisms, we still call them mesophiles because it's close to 37 degrees where their optimum temperature is. Any question about any of that? All right, and I guess if you're looking at some microbes growing in the Antarctic or the Arctic, they're growing below 0 degrees, and that might be their optimum temperature for growth, as opposed to the refrigerator, the organisms growing in there, and that optimum would be, around 10 degrees. It may be a little cooler depending on how cold your refrigerator is, but it would always be above 4 degrees Celsius. And I think always below 12 degrees Celsius. Any question about any of that? All right, we talked a little bit about food spoilage. Let me mention it a little further here. Food spoilage occurs in Celsius between 52 degrees when the organism is growing in the food, and the food is between 52 degrees Celsius and about 15 degrees Celsius. That would be about 60 degrees Fahrenheit to maybe 130 degrees Fahrenheit. Rapid growth of bacteria. The bacteria may grow and produce some toxins. So after about 4 hours at this temperature, the food should no longer be kept. Now, obviously, 60 degrees Fahrenheit is actually room temperature, or a little below room temperature. And food spoilage can occur at a range below that temperature. Generally, we say that bacteria can survive. Some may actually grow between about 5 degrees Celsius and 62 degrees Celsius, although the bacteria are growing much slower here. At the refrigerator temperature, the bacteria should stop mesophiles from growing, but the bacteria in the refrigerator itself can grow, and it can cause spoilage of the food. But there are very few pathogens that grow at that temperature in the refrigerator. Food spoilage can occur when the food cools too slowly, like when you have a very large vat of food and you put it in the refrigerator. The food cools too slowly, and so the bacteria has enough time to grow and can perhaps make toxins, which can then make people sick just between, I don't know, well, let's say 8 hours. The food is in the danger range for 8 hours before it's put in the refrigerator. And then when it's put in the refrigerator, it's cooling too slowly. So you need to put it in a smaller vat and then have the temperature drop quicker so that it drops below about a 15 degrees Celsius under 2 hours. And then the bacteria cannot grow and produce toxins. Any question about any of that? Oops. Another physical requirement is the pH that an organism requires to grow at. Most bacteria grow best between 6.5 and 7.5 in pH, so around pH 7. And in lab cultures, that's usually what we give you. If you were to come into a lab and had a media, the media would be a pH around 7. However, when the bacteria grow in the media, the bacteria can produce acids, and the acids may inhibit growth. To minimize the pH change in the media we give you in the lab, we use buffers, pH buffers, and they reduce the change in the pH, reduce it so the pH doesn't swing dramatically, like when microbes are growing in the media. Molds and yeast can grow at a lower temperature, though, and they grow best between pH 5 and pH 6, so slightly acidic. Acetophiles grow in pHs less than 7. Anything less than a pH of 7 is called an acid, and acetophiles grow in acidic environments. Some of them can grow in volcanic environments where the pH will be between pH 0 and pH 3. The only thing we have that low in pH would be our stomach. And yes, microbes can grow in your stomach, and those would be acetophiles. Helicobacter pylori is a bacteria that can grow in your stomach, and actually it can cause ulcers, and it can grow in a very acidic environment. Now, the pH of the stomach is usually between pH 1 and about pH 2. It doesn't ever get down to pH 0. There are other physical requirements, such as osmotic pressure. Microbes require water in order to function. If there isn't enough water around for the microbe cannot engage in metabolism. That doesn't mean the microbe will die. Most microbes will die, like E. coli, if you have it in a desert environment or an environment with very low water. The water will leave the microbe and then the E. coli would die. However, there are some microbes which resist desiccation and would resist dying in a very dry environment, such as species of Mycobacterium that have mycolic acid in the cell wall that keeps the water inside the cell from leaving the cell. Another example would be an endospore. They can survive in dry environments for decades or even millennia. High osmotic pressure can remove necessary water from a cell. The cell loses too much water, metabolism cannot occur, and then the cell will die. In a hypertonic environment, the solute concentration is higher outside the cell than inside, so water will leave the cell. Let me blow that up. And then the cell membrane will shrink from the cell wall. We call that plasmolysis. That generally inhibits growth if it does not kill the cell. Like E. coli would quickly die if this were to happen. Like you put E. coli in 10% sodium chloride, it will lose water and then will quickly die. Hypertonic environments can be when there's more salt or sugar outside the cell. Actually, any solute outside the cell. There's more of it, more of it outside the cell than inside the cell. That's high osmotic pressure, and it can be used to preserve food. If you don't know, a bottle of molasses can be kept at room temperature for years. My mother had a bottle of molasses that she wasn't using, and we figured it was about 20 years old. And I took the molasses and made my father molasses candy, and he ate it, and it didn't kill him. He enjoyed it. And that bottle lasted about 10 years longer. And then we realized that this molasses was at least 30 years old. At that point, my mother decided, I think it's time to throw that bottle away, which she may as well because she wasn't using the molasses, because that's why it kept around that long. She never used it. My point is, when the cell fell into the molasses, rather than digest the molasses, the water left the cell, and then the cell either could not grow if it was an endospore, or it died if it was something like E. coli from plasmolysis. Cells generally prefer an isotonic solution. That's actually, I'm not sure why that's 0.85. It actually should be 0.09% is isotonic. It might be that bacteria prefer a little less salt. Let me think about that. No, they don't. They don't prefer a little less salt than our cells. So I'm not sure why that's showing 0.085. Should be 0.09% is isotonic. And cells generally prefer that because then we don't have water either entering the cell or leaving the cell if the cell is in isotonic solution. Halophiles, or organisms that like being around salt. Halo means salt, phile means lover, so these means a salt lover. There's two types of halophiles. There's an obligate halophile, and they require a high salt concentration in order to grow, like the organisms that live in the Dead Sea between Jordan and Palestine. It's about 30% sodium chloride. Any microbes in that water have to be obligate halophiles. And then there's a facultative halophile. They do not require high salt concentrations to grow, but they can tolerate salt at higher than 2% sodium chloride. Some can grow in 15% sodium chloride environments. An example of facultative halophile would be Staphylococcus aureus. it can grow on tryptic soy, agar, or any general purpose media, which is only isotonic, meaning about a 0.9% sodium chloride. So it does grow in that. But it can grow on your skin, which can get up to 20% sodium chloride. And it tolerates that high salt. I'm not sure if it can grow in 20% sodium chloride, but the bacteria can tolerate it and will survive the 20% sodium chloride of your skin. Now, obviously, how salty your skin will be depending on when you had your last shower or bath and how much you've been sweating. But the human skin can get up to 20% sodium chloride. And Staphylococcus aureus can grow on your skin. So it's a facultative halophile. Doesn't require it, but it can tolerate it. In a hypotonic solution, in low osmotic pressure, meaning there's more water outside the cell than inside the cell, water will enter the cell by osmosis. This may cause the cell to swell, and if the cell does not have a cell wall, the cell membrane will expand and expand until it bursts. If the cell has a cell wall, which most bacteria do, and then green plant cells do, the cell wall will prevent the cell membrane from expanding any further. And then water can no longer enter the cell because there's no room for the water inside the cell, because the cell membrane will not expand. Any questions about halophiles? All right, another chemical requirement is carbon. All living things have to have a carbon source. The typical cell has half of the dry weight of a cell being carbon. So if you take your cells and remove all the water, half of the weight will be from carbon. Carbon is used as in structural organic molecules, meaning your cells have structure because of carbon. Carbon is also an energy source for cells, like we will use the carbon in glucose to obtain most of our energy. The carbon comes from glucose. Heterotrophs use an organic carbon source. An autotroph can get their carbon from CO2, either in the air or the water. Any question about any of that? Some other chemical requirements include nitrogen, sulfur, and phosphorus. Nitrogen, about 14% of the dry weight of a cell, is from nitrogen. Nitrogen is in amino acids, proteins, DNA, RNA, and ATP. Many cells obtain their nitrogen from decomposition of protein. And that's how our cells get their nitrogen. Some bacteria can digest protein, but others can get their nitrogen from either ammonia or ammonium, NH4, or from nitrate, NO3 with a negative charge. We cannot. Green plants can, though, especially nitrate. They love nitrate, green plants. A few bacteria can even get their nitrogen from nitrogen gas, and these we call nitrogen-fixing bacteria and archaea, because they can pull nitrogen from the atmosphere and then bring it down into the ecosystem. Initially, they'll convert that nitrogen into a nitrogen compound like amino acid or a protein, a biological form of nitrogen. The point is that nitrogen can then cycle around in the ecosystem. Sulfur We get ours from amino acids. If you take thiamine or biotin, those are B vitamins. They're also a source of sulfur. We need sulfur for DNA and RNA synthesis. Most sulfur comes from the decomposition of proteins. Our cells get most of their sulfur from decomposing proteins. But many bacteria can get their sulfur from protein, but some get it from sulfate or hydrogen sulfide gas as their sulfur source. We cannot. Hydrogen sulfide gas is actually toxic to us. Phosphorus is found in DNA, RNA, and ATP. It's also found in phospholipids of the cell membrane or any membrane in our cells. Phosphorus is usually found as phosphate, PO4 with a 3 negative charge. So that's an important source of phosphorus. And we can get this from our diet, like meat or milk. Important source of phosphorus for us. Phosphate is usually the form that most microbes use. And we can actually use phosphate too. Trace elements are inorganic elements required by microbes in small amounts. They're usually picked up from their environment. And that's true for us too. We get trace elements from the food we eat or the liquids we drink. Trace elements may be required as an enzyme cofactor. Some example of trace elements include iron, copper, and zinc, elements that we need in trace amounts in order to stay healthy. Any question about any of that? Like a good source of copper would be to eat grapes, but I think any dried fruit would be a good source of copper. Fruit would probably be a source of copper, it's just that it wouldn't be highly concentrated. If you remove the water and have dry fruit, you'll get more copper. Iron, you can get that from red meat, from broccoli. If you cook in a cast iron pan, you can get it that way. I've forgotten some of the other sources of iron, but those are some sources. I think green leafy vegetables tend to be a source of iron, which is why broccoli is a good source. Organic growth factors are another chemical requirement required by certain organisms. Organic growth factors are organic compounds obtained from the environment that the organism cannot synthesize itself. Some examples for us would be vitamins. Vitamin C, we cannot make it. We must get it from our diet, or maybe from our liquids we drink if you drink orange juice. Certain amino acids are organic growth factors. These would we call essential amino acids, like lysine. We cannot synthesize it, so we have to get it from our diet. And then essential fatty acids too. Some organisms require purines and pyrimidines as a organic growth factor, and you need these to make a DNA and RNA. Now, how we get most of our purines and pyrimidines is we will digest DNA or RNA, which will be found in cells. And when we eat those cells, we will digest the DNA and the RNA. Oxygen is another chemical requirement. Molecular oxygen is, in a sense, a toxic substance. The reason is that molecular oxygen can be metabolized into other forms, which can be reactive and then toxic to cells. And that includes aerobes. So these other forms of oxygen, the toxic forms, are toxic to even aerobes like ourselves. Any question about that? Oxygen is, of course, toxic to an anaerobe because an anaerobe doesn't want to be around oxygen, especially a strict anaerobe. Oxygen would be toxic to it, and the anaerobe would not grow and may die when exposed to oxygen. There are 4 toxic forms of oxygen. Oxygen gas, at least to us, because we're an aerobe, is not toxic. But there are 4 toxic forms of oxygen. One is singlet oxygen, where oxygen gas is boosted to a high energy state. The oxygen is extremely reactive. Singlet oxygen is actually present in phagocytic white blood cells, and then in the lysozyme of the phagocytic white blood cell. And then the lysozyme can fuse to whatever the white blood cell is attacking, and then help kill what the white blood cell wants to kill, the singlet oxygen. The second form of a toxic form of oxygen is superoxide free radicals. This is where we have oxygen gas with an extra electron, which is O2 with a circle or square, and then a negative charge. The negative charge is because the electron has a negative charge. Superoxide free radicals is an unpaired electron. We only find unpaired electron in free radicals. These molecules are highly unstable. They are formed during normal aerobic respiration. And when this molecule, the oxygen superoxide free radical, bumps into another molecule, it'll steal an electron from that molecule because that unpaired electron wants to be paired. And it can damage that molecule it steals the electron from. If it hits DNA and steals the electron from DNA, it can mutate the DNA. Superoxide free radical is toxic to even our cells, and we have an enzyme, superoxide dismutase, or SOD, which takes superoxide radical, 2 of them, combines them with 2 hydrogen ions to make hydrogen peroxide and oxygen. And that's less toxic than superoxide free radicals. You'll note that hydrogen peroxide is toxic to our cells, but our cells have a way to solve that problem. So SOD changes one toxic form of oxygen to another toxic form. And then that's our next third form of a toxic molecule, and that's a peroxide anion, such as hydrogen peroxide. 2 peroxide anions can combine with 2 hydrogen ions, forming oxygen hydrogen peroxide. Hydrogen peroxide is a peroxide anion. Well, it's a peroxide molecule. The hydrogen peroxide is then neutralized by an enzyme. In our cells, hydrogen peroxide is neutralized by the enzyme catalase, which converts 2 hydrogen peroxide into 2 water molecules and oxygen. If a cell does not have catalase, it can convert hydrogen peroxide into water by the enzyme peroxidase. And that reaction is a little different. You get 1 hydrogen peroxide plus 2 hydrogen ions in the presence of peroxidase enzyme to make 2 water molecules. All bacteria that live in the air have to have either catalase or peroxidase, or maybe both, to detoxify hydrogen peroxide. Our cells have catalase and only catalase. Any question about any of that? And if you ever wanted to test whether your cells have catalase, you can put hydrogen peroxide on a wound or cut, and you'll see the bubbling. That's from the catalase converting the hydrogen peroxide into the water and the oxygen. The oxygen creates the bubbling. The 4th toxic form of oxygen is a hydroxyl radical. It is similar to the 2nd form, which is the superoxide free radical, where oxygen has an unpaired electron. However, here it's actually not oxygen, it's OH with an unpaired electron. Note it is not OH minus the hydroxide ion. It is OH with an unpaired electron. OH hydroxyl radical is formed in aerobic respiration in trace amounts. It's also formed by ionizing radiation and when hydrogen peroxide reacts with certain metal ions, like in your cells. Hydroxyl radicals are toxic to our cells, and they will bump into another molecule, stealing an electron and potentially damaging the cell. When a hydroxyl radical bumps into DNA, it can mutate the DNA. Our cells have antioxidants that remove this and other free radicals. One such antioxidant is actually vitamin C. Another would be vitamin E. These are antioxidants that combine with hydroxyl radical to remove the hydroxyl radical from our cells. Unfortunately, that does remove the vitamin C or the vitamin E from our cells as well, meaning our cells cannot reuse that vitamin C, but it does remove the free radical from our cell. Any question about any of that? All right, microbes that use oxygen we call aerobes because they require the growing in air or else they'll die. They require oxygen. And I suppose you could grow them in pure oxygen if you wanted to, but cells are used to 20% oxygen, which is what we have in our air. Aerobes, remember, they make more ATP than anaerobes. And so aerobes do grow faster than anaerobes. They make more ATP from whatever molecule they're digesting, which in most cases would be glucose. Microbes are classified on the basis of the effect oxygen has upon their growth. We have aerobes, which grow at the top of a thioglycolate tube. Thioglycolate is a media that removes oxygen from the media. But once you take the tube out of the autoclave, the oxygen comes in from the air, because this tube is not sealed, oxygen can come in. And then it will make the thioglycolate media at the top full air, or around 20%. And as you go down the tube, you'll have less and less oxygen until you reach the bottom of the tube, and that will be no oxygen. And it's because thioglycolate removes the oxygen, and then oxygen comes back into the tube and then creates an oxygen gradient. At the bottom of the tube, we have no oxygen. A little higher up, slight oxygen. A little higher up, a little bit of oxygen. A little higher up, some oxygen. And then at the top of the tube, full oxygen, or around 20% oxygen. An aerobe grows only at the top of the thioglycolate tube. And let me see if I can blow this up. There we go. You'll note that there's more growth at the top of the tube, and as you go down the tube, there's less and less growth. I should put a dot right about here to show there's a little bit of growth right in that region here. And that's because there's a little bit of oxygen, so there will be a little bit of growth about midway through the tube. Any question about that? The point is that we'll have most of the growth at the top of the tube, and as you go down the tube, you'll have less and less growth. The next easiest one to talk about is an obligate anaerobe. They require the absence of oxygen in order to have growth. And these, we would find the most growth at the bottom of the tube. As you go up the tube, there will be less and less growth because there will be more and more oxygen. Once again, I should probably put a little dot right about there, because about this region, there will be slight growth of the bacteria because there's oxygen, but the oxygen isn't everywhere. And so the microbes can grow a little bit right here, because there's some regions that are without oxygen. When you go up the tube, there will be no growth because oxygen will be found at higher and higher concentration. And these guys are obligate anaerobe. They will not grow in the presence of oxygen. The next is the facultative anaerobes, and they can grow everywhere in the tube. What we should do is probably have one dot right here, and then as you go up the tube, there's more and more growth. We can see the most growth here, and there's more growth there because that's where there's most oxygen. Facultative anaerobes can perform both aerobic respiration and anaerobic respiration. They grow best and fastest where there is oxygen, and they grow best where the oxygen is the highest. As you go down the tube, there will be less and less growth because there will be less and less oxygen, and they'll have their greatest growth when they're performing anaerobic respiration. But we'll have growth everywhere in the tube because these organisms can grow anaerobically if oxygen is not around. So if there is no aerobic respiration going on, they'll perform anaerobic respiration at a slower rate. They'll grow slower, while you'll see slower growth the further down you go to the tube. At the bottom of the tube, you'll still have a little bit of growth from anaerobic respiration. Any questions about facultative anaerobes? All right, the next anaerobe we need to talk about are aerotolerant anaerobes. These are anaerobes, so they are not using air or oxygen to grow. They are anaerobes, and they grow without using oxygen. However, these organisms, the aerotolerant anaerobes, have all of the enzymes they need to detoxify the toxic oxygen molecules. So they can grow anywhere in the cell or anywhere in the tube at the same amount. So at the top of the tube, we have some growth. In the middle of the tube, we have some growth. In the bottom of the tube, we have some growth. And the growth will be the same throughout the tube. And it's because they have the enzymes to detoxify the oxygen toxic molecules. So if oxygen is around, it's not a problem. They still grow at their optimal rate. If there is no oxygen around, it's no problem because they don't need it to grow, and then they grow at their optimal rate. All right, there's a fifth one I need to talk about, and it is microaerophiles. They are aerobes, so they do require oxygen to grow. However, they do not like growing at full oxygen. The oxygen is too high for them, and so they do not prefer growing at that temperature, or that oxygen concentration, sorry. Where they grow the best is in the middle of the tube, where the oxygen concentration is something like 2 to 8% oxygen, and that's where they'll have their optimal growth. As you go up the tube, there will be less and less growth, and as you go down the tube, there will be less and less growth. When you're going up the tube, there's less growth because oxygen is becoming higher and higher in concentration, and the cells don't like growing in that high of oxygen. It doesn't mean they won't grow. There will be less growth as you go up to the top of the tube. And as you go down the tube, there will be less growth because they are aerobes. They require oxygen. And as you go down the tube, there will be less and less oxygen. So the cells will grow slower. At the bottom of the tube, there will be no growth because there's no oxygen. The very bottom. But I should have a dot right about here and a dot right there to show that we do have growth, less growth around the middle of the tube. Those are microaerophiles. You do have a microaerophile in your stomach. The stomach oxygen is generally between pH 2 and pH 4, and Helicobacter pylori is a microaerophile. Any question about any of that? And that's the bacteria that grows in your stomach. Why the oxygen is at around between 2 and 4% is the oxygen gets into your stomach, like when you swallow, oxygen is brought down, especially if you're swallowing air. But we have microbes in our stomach which use up that oxygen, and that lowers the percentage of oxygen in the stomach to 2 to 4%. Not all microaerophiles grow best at that oxygen concentration, but Helicobacter pylori does grow best at that oxygen concentration. Any question about thioglycolate tubes and the effect of oxygen on microbial growth? If not, let's talk about culture media in general. Culture medium is singular. So when we're talking about one, you should use the word medium, or the medium that I picked up today, for example. However, if you pick up 2 or more, then you call it media, because media is plural for medium. So if I pick up 2 or more media, like a general purpose media and I don't know, 7% sodium chloride media, that would be 2 media. Media can be liquid or solid solution containing all of the nutrients required for microbial growth. The media you get in the lab is usually sterile, meaning they have no living microbes inside them. How they're sterilized is you put them in the autoclave. An inoculum is the microbes introduced into the medium to grow. The culture is the microbes growing in or on the culture medium. So a culture would be what we're growing, and it's the cells growing in the culture medium that you have the cells growing in. There are a very wide variety of media that we have available in microbiology. Let me see if I can show you. We have a book that's actually a little bigger than this one, but it's smaller than that one. Is that bigger? No, it's a little bigger than this, a little wider, maybe a little bit longer too. On in each page of this book in our lab, it has one media. This book is about that thick. So it has a very wide variety of media that we have available in microbiology to grow a culture in. Any question about any of that? And yeah, we'll have to talk about each of those media, then each of those pages. I should look up how many different pages it is, but I've never done it. The book's about that thick though. One page for each media. Culture media is used to support microbial growth. The media must provide an energy source, a carbon source, a source of nitrogen, sulfur, phosphorus, trace elements, and organic growth factors if the organism needs them. Like if you were growing our cells, which isn't a micro, but you would have to supply organic growth factors like vitamin C in the media for our cells to grow. E. coli does not require any organic growth factors. It does require trace elements, but you usually get that from the water or the substance that you make to make the media. Like if you ground up the cow will have trace elements, and you can use that. E. coli will require a source of nitrogen, sulfur, and phosphorus, so that has to be supplied, a carbon source, and an energy source. But let's say we're trying to grow a photosynthetic bacteria, and then we're giving them light. You don't need to supply an energy source. Do you need to supply a carbon source for a photosynthetic organism? You guys should know this.
If you're in space, you do.
Photosynthetic microbe. Say again.
If you're in space, you do.
Let me think about that. You can't grow cells in the vacuum of space. So if you had it in the spaceship, you would have you'd have carbon dioxide in the air, and the photosynthetic organism most likely would be able to get CO2 from the air. So even in space, at least in the spaceship, you're supplying the carbon source from the air, because most photosynthetic organisms can utilize CO2 from the air. There's only a few that could not. We're not going to talk about them. In which case you would have to supply a carbon source if it did require a carbon source. Like E. coli, we do have to supply a carbon source for growing that cell. And the carbon would usually be from something like glucose or proteins in the media. There has to be a source of nitrogen. Like for E. coli, you can supply protein to supply the nitrogen. Protein will supply the sulfur. For phosphorus, you have to supply phosphate, which is usually added to the media. I guess you could grind up the cow, and then the ground up cow can supply the phosphate. You do have to supply trace elements. That would be things like iron, zinc, magnesium. Generally, you'll get that from the water if you're using tap water, or if you're using a media and you're grinding up the cow, the trace elements will come from the cow as well. If the organism requires organic growth factors, you'll have to supply them in the media. Like Streptococcus pyogenes requires organic growth factors or it will not grow. So it will not grow on tryptic soy agar, but it will grow on blood agar because the red blood cells are supplying the nutrients that Streptococcus pyogenes requires in order to grow. Any question about any of that? All right. I think I stated that the media could be liquid or it could be a solid. We often use agar to solidify a liquid media to make it a solid media. So agar is a solidifying agent. It's a complex polysaccharide derived from seaweed. We usually have agar in a petri dish, but it can be in a tube. We have two types of tubes. Oh, there we go. A deep and a slant. So we have a tube like this, and the agar just runs across like that. This is a deep. On the other hand, if we have a tube like this, and the agar is on a slant, this we call a slant. Any question about any of that? So two different types of tubes with agar, a deep and a slant. Don't think I'll need that, but I'll keep it just in case. Agar is generally not metabolized by microbes. Why we use it so frequently in microbiology when we want a solid medium. You do not have to use agar though. You could use gelatin or you could use starch to solidify a liquid media and make it solid. One problem with gelatin and a little bit less of a problem with starch is that microorganisms can digest both gelatin or starch. And I suppose if you don't care and all you want is growth, it's fine. But if the microbe starts digesting the agar, you'll no longer have a slant. You'll start digesting the slant, and then we'll have the agar become liquid. And so we'd have something like this, a liquid layer over the solid layer. So that would be solid right here. And here would be the liquid. And usually we wouldn't want the bacteria to liquefy the agar because your cells and colonies would then mix together. And that's why we use agar, because the bacteria cannot digest the agar. We have different types of culture media. There are chemically defined media where the exact chemical composition is known. And why it's known is because you put all the chemicals in the media. So you know everything that's in the media. Let me blow this up. Here's a simple chemically defined medium. It has glucose, ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, and then just water, H2O, 1 liter. And those are the only molecules in this media, assuming you're using distilled water. Obviously, if you're using tap water, you're going to supply trace elements. But if you use distilled water, you know what all the molecules are because you put all of them in the media. And this will be a liquid media. You'd have to add agar to make it a solid media. The exact chemical composition of a chemically defined media is known. These are usually used for growing autotrophs because they do not require a large number of additives to the media. Oh, this has glucose in it. That's not exactly what you would put in for an autotroph to grow. This looks like it's glucose medium. Supplying the correct osmolality, giving nitrogen, phosphate, magnesium, and sulfate, and then more phosphate as well as potassium. And then glucose would supply the energy and the carbon source. So there are some microbes that could grow in this media. I think E. coli grows in glucose media. A lot of cells would not grow in glucose media. And obviously Streptococcus pyogenes would not. But there's many others that would not, but I'm not calling to mind any at the top of my head right now. I used to know this because I used to supply my students with glucose media, but it's been many years and I don't remember what cells grow in glucose media and what don't. I could look it up, but I'm not going to take the time to do that now. Like I said, chemically defined media is usually used for growing autotrophs because they don't require a lot of additives. A fastidious organism, on the other hand, requires a lot of additives. Oops, let me bring that back. Just blow it up. What I'm trying to do here. So here is a chemically defined media, which has a bunch of the things above. Oxaloacetate's a little different, but it's providing a bunch of different minerals and nutrients here. All of these are fairly normal. Calcium chloride, iron source, nitrate source. But here are some, like this is giving cysteine to the media, arginine, proline, glutamic acid, methionine, asparagine, isoleucine. So a bunch of amino acids are provided. A bunch of organic growth factors are provided, including the B vitamin there. And then this is added sodium thioglycolate because this is for growing a anaerobe, an organism that doesn't like exposure to oxygen. So we have sodium thioglycolate to remove oxygen from the media. The point is, this has a lot of additives you have to add because this is a fastidious organism that requires all of these additives. Like thiamine and biotin, those are B vitamins. I don't remember what that is. Nicotine adenine dinucleotide, that's NAD. NADH probably. I'm trying to check if that's NAD plus or NADH. I think that's NADH. So fastidious organisms require a lot of additives. Usually we don't do this. We just add something like red blood cells, and then they provide the fastidious organism with the additives that it needs. And I don't know what red blood cell provides, but it's obviously providing whatever Streptococcus pyogenes requires to grow. There's also complex media. These media contain extracts or digests of yeast, meats, and plants. These provide vitamins and organic growth factors. The exact chemical composition of a complex media is not known. Let me blow this up now. Like for this complex media, where we're adding peptone, that is a partially digested protein that you get from soybeans. Beef extract, so 3 grams of ground up cow, and I should say dried cow too. Sodium chloride to give it the right osmolality, the right pH to make it isotonic, and then just agar to make it a solid media, and then water. So really, this one only has 3 things providing nutrients, and sodium chloride is not much nutrients. Peptone, and then beef extract, and then sodium chloride. How we're getting all the nutrients, like the carbon source, the nitrogen source, the sulfur source, any trace element is from the beef extract. Grind up the cow. That's especially true if you're not getting trace elements from tap water, that you're using distilled water for your water. So the trace elements will come from the beef extract. Perhaps from the peptone digest too, depending on how purified they have that for the soybeans. The point is that the cow is going to supply of different molecules, a lot of different molecules. Depending on what the cow is eating, the cow will have that molecule. And the exact composition of that media will never be known because nobody checks the cow to what molecules it has inside of it. And then different batches of the media will differ because different cows have different molecules in them, depending on what they're eating. Like in Oregon and Washington, we tend to feed the cows grass. But in, I'm trying to remember that desert plant that they feed the cows in, sage maybe, or sagebrush, something like that, is what they feed the cow in like Colorado and other places where it's very dry, like Grand Junction, Colorado. And then in the South, you feed the cow a bunch of corn. And so the cow will have different molecules than if the cow is eating grass. We usually use complex media when we're growing heterotrophs, like heterotrophic bacteria that require an organic carbon source. And then it gets that whatever it needs from the peptone extract and the beef extract. We also have anaerobic media. This is important for growing obligate anaerobes. The anaerobic media removes oxygen from the media. We call that a reducing media. It contains a chemical that removes oxygen. In the case of sodium thioglycolate, that chemical will literally combine with oxygen and remove oxygen from the media. And then you can grow a strict anaerobe, at least in the bottom of the tube. Obligate anaerobes must avoid exposure to oxygen because oxygen will kill them. And in the thioglycolate, how you drive off the oxygen is you autoclave it. That drives off the oxygen from the media. But once you take the tube out of the autoclave, oxygen begins adding to the media, and the top of the thioglycolate tube will be full oxygen. As you go down the thioglycolate tube, you get less and less oxygen. So there'll be no oxygen at the bottom, less oxygen halfway, and then full oxygen at the very top of the thioglycolate tube. Any question about any of that? Another type of media we have is selective media. This media has a chemical in it that halts or inhibits the growth of a particular group of microorganisms while not affecting the growth of another group of microorganisms. An example of a selective media is 7% sodium chloride media. This media has 7% sodium chloride in it, which prevents most microbes from growing. Most microbes can grow, I'm not sure what the low number is, but someplace around 0% sodium chloride to up to about 2% sodium chloride. Beyond that sodium chloride, most microbes do not grow because there's too much salt around. And 7% sodium chloride media has 7% salt around. So only the salt lovers can grow. We call those halophiles, like Staphylococcus aureus, a halophile. And then it inhibits the growth of other organisms, like E. coli, cannot grow on 7% sodium chloride media, but Staphylococcus aureus can. That's selective media. There's also differential media, and this media provides differences between different groups of cells, showing you differences between the two groups. The differences are not related to how well the organism can grow on the media. The differences do make it easy to distinguish colonies of different microbes. So one differential media can make one cell a certain color and another cell a different color. Differential media. Here we're looking at blood agar, and it's differential for the type of hemolysis. Let me blow this up a little bit. Oh, that's good enough. So white, we have cells growing right here. These cells are showing gamma hemolysis, also called no hemolysis, because the red blood cells are present all the way up to where the cells are. No hemolysis, called gamma hemolysis. And that's the shape of a lower gamma in delta in Greek alphabet, by the way. This is alpha hemolysis, which is partial hemolysis. Let me blow that up a little bit more. What you see is a dark ring. The off-white color is the bacteria. And then we have a dark ring of the blood. Very thin ring, but there is a dark ring around the cells. And the biggest place to see it is inside the alpha right there, how that's darker than the red here. The alpha hemolysis is usually seen as darkening of the blood or as greenish coloring of the blood. Here it looks like it's darkening. And that's partial hemolysis, called alpha hemolysis of the blood. We also have beta hemolysis shown here. The bacteria is in the white beta right here. And then we have a clear region right there, and that is beta hemolysis being shown there. It's also called complete hemolysis, where all of the red blood cells are lysed. White is clear. And you can actually see right through the plate right here. You can hold this plate up to the light, and you can see the light bulb behind the plate. Or if you hold it up to the clock, you can see the clock through this portion of the plate where the beta hemolysis is. Can't see it through where the bacteria are or wherever there's red blood. Any questions about blood agar and the different forms of hemolysis? I think I mentioned before, around beta hemolysis, you always have a ring of alpha hemolysis. So that's normally seen. Now, we said there were selective media and differential media. There's also selective and differential media. EMB, or eosine methylene blue, is such a media. It is selective in that it selects against the growth of gram-positive bacteria from growing on the plate. So if the bacteria can grow on the plate, it's likely to be gram-negative. I'm saying likely because the selection isn't perfect, and it's true in about 95% or greater of cases. Gram-positive will not grow on EMB, and gram-negatives will grow on EMB. But there are a few exceptions. It is selective because gram-positive do not grow, but it is also differential. And it's differential on the basis of lactose fermentation. Lactose is added to the EMB media, and the bacteria will be blackish in color, shown here, or greenish in color, shown here, when lactose fermentation occurs. And that produces an acid, causing the plate to be either black in color, or the cells to be black in color, or greenish in color. If the bacteria do not ferment the sugar lactose, and the bacteria can grow on EMB, then the bacteria will be their normal color, which in this case is either white or off-white. Let me blow that up so you can see that a little better. So that colony is white. The streak is white. You cannot distinguish the differential ability of the cells if they do not grow on the plate. So if the bacteria is gram-positive and does not grow on the plate, you cannot distinguish whether the bacteria ferment lactose or they do not. Any question about any of that? Now, I mentioned that blood agar is differential. It is also an enriched media, and that is it allows fastidious, or at least some fastidious organisms to grow, like Streptococcus pyogenes can grow on blood agar, but would not on tryptic soy agar, because Streptococcus pyogenes needs additives that must be supplied by the media, and the red blood cells are supplying the additives. So I've talked about here blood agar being differential, but in reality it's both differential and an enriched media. Now don't confuse enriched media with an enrichment media. An enrichment media encourages the growth of certain organisms to grow. So an enrichment media is selective because it's selecting for what it's encouraging to grow. But we don't use it for the same purpose we do for a selective media. We use an enrichment media to increase the numbers of a desired microbe to detectable levels. It's particularly useful if you're targeting a microorganism that's only a small proportion of the total population, such as a microbe in the soil or a fecal sample, like a patient has diarrhea and you're doing a sample and you're hoping to determine what might be causing the diarrhea. Well, there's so many bacteria in the feces, you might not find the one that's causing the diarrhea because it's in low numbers. So what you could do is put your fecal sample, put the bacteria from it in an enrichment media that's selecting for a human pathogen. Like suppose you suspect it's salmonella, you can put it in an enrichment media that selects for salmonella. And then if you get growth in it, you can then streak it out, and then you'd have a chance of obtaining a colony that's streaked out twice to get a pure culture that would be causing the pathogen. So let's say salmonella is only 1 in 1,000 cells in the feces. If you were to streak out a plate and you got 10 isolated colonies on the plate, and then you tested each of those 10 isolated colonies, you're not likely to find salmonella simply because salmonella is not likely to be one of the 10 colonies, because to get one colony that would be salmonella, you'd have to sample 1,000 different colonies. And in one plate, that would be impossible to do. But if we were to grow the fecal sample in an enrichment media that were to enrich for salmonella, we could increase the concentration of salmonella from, let's say, 1 in 1,000 to 1 in 10. Now, if we streak out a plate and have 10 isolated colonies, at least one of them would likely be salmonella. And that's what we use an enrichment media for. Any questions about any of that?
Yes, I have one question. I just want to make sure that I heard you correctly. So you're talking about trying to isolate one particular colony, and you mentioned needing to streak it out twice. So my understanding is that we streak out the first plate, but there's still too many colonies, so we need to streak out a second plate in order to isolate for, and then can we still isolate a pure culture on the second one then?
No, you got that wrong. I talked about this before, and that is to get a pure culture, and it won't, excuse me here, I had a frog in my throat. If you were to streak out isolated colonies one time on a plate, only about 70% of the colonies would be pure. And that's because the colonies can be formed by more than one cell. And if a clump of cells form the colony, you're likely to have that colony a mixed culture, meaning there's more than one species in that colony. So what you do is you take from an isolated colony on one plate and then streak it out on a second plate to get a second isolated colony. You've now taken the cells through 2 colony formation events. This will increase the chance that you'll have a pure culture to 95% of the colonies or higher. Do you see?
Yes, thank you so much for explaining that one more time. I really appreciate it.
Okay. And so that's why you streak it out twice, just to make sure you get a pure culture. And in reality, you're not going to get a pure culture 100% of the time, but it'll be less than 5% of the time you'll get a mixed culture, even if you streak it out 2 times. And in all my years of teaching, I've never had a student streak out their culture twice and then they had a mixed culture that was not pure. Okay. Every time I've found a student who thought they had a pure culture, they only streaked it out one time. And then they did a gram stain and they were seeing two different cells on it, which is lucky the gram stain showed two different species, because if they had been both like two gram negative bacilli, the gram stain isn't going to show it. But it happened to be, I don't know, either a cocci and a bacilli, or one was gram negative and one was gram positive. And then I asked the student, well, did you streak this out 2 times? They said, no, I only streaked it out 1 time. And so you never want to streak it out 1 time, because 30% of the time is way too high to assume you have a pure culture. It's 30% of the time it will not be pure. So you need to streak it out 2 times. All right, this is just giving you an example of enriching from a phenol degrading bacteria you want to find in the soil. Phenol degrading bacteria are very rare in the soil. It might be 1 in a million cells. There would be no way to plate the bacteria from the soil on a plate, streak out colonies, and get 1 colony that would be a degrading, phenol degrading bacteria. So what you would need to do is plate it in an enrichment media that would select for, let me see if I can find that. There we go. Select for phenol degrading bacteria. And what you do to get that media is you simply have the only carbon source, phenol, in the media. That will select for phenol degrading bacteria. So initially we had 1, I don't know if I can write this in down here, 1 times 10. Well, let's just say 1 and 10 to the 6 cells. I'm just going to go 1 over 10 to the 6 cells. I don't know if I can do a superscript here. Not that I see, so let's just go EE. This is the old way of doing it on a computer, by the way. Come on. And let's say this phenol degrading bacteria increases the enrichment by 100 times. That would only increase it 1 times 10 to the 4th. But what you can do is, oops, I'm at the wrong one. What do I want here? This out of the way. Put it in another enrichment media. So transfer it to here. And it was 10 to the 4th in this one. And then we enrich it here, 10 to the 2. So instead of 10 to the 6 now, it's now 10 to the 2. And that would be after you really streak it into another one here. So that'd be 10 to the 4th. This would be 10 to the 2. I'm just going to go 1 over 100. We could streak out 100 colonies. That's a little high, but we could do it 1 more time. You do it 1 more time, you're going to have a very good chance of finding the cells, because if you have the same chance, it would be close to 1 out of 1 here. Oops, don't have the thing. It'd be close to 1 out of 1 here. And then your chance of streaking out a phenol degrading bacteria would be very high. This would be high enough to even get it. You have to streak out 100 colonies, but that's doable. right, any questions about any of that? If not, I'm going to stop here and I will see you at, let's make it 7:02 for the lab because I'm a little late here. Sorry, I went a little long here. All right, any questions? All right, I'll see you at 7:02.